Good afternoon. My name is Karen Howard, and I'm the CEO of the Organic and Natural Health Association. It's a trade association comprised of consumer organizations and corporations committed to preserving and advancing the health and well-being of people, animals, plants, and the planet as a whole. Led by our guiding principles, sustainability, transparency, accessibility, and traceability, we are embarking on a series of projects, including our focus for today, facilitating scientific research on ingredients and products and the impact they have on healthcare status. This effort is being driven through our partnership with GrassrootsHealth, a nonprofit consortium of scientists, institutions, and professionals committed to worldwide nutrient sufficiency that has pioneered nutrient research by using sophisticated field trials. Before I introduce our first panelist, let me first address a few housekeeping items. All participants are more than welcome to submit questions via the chat function. We will be taking questions at the end of the session, time permitting. The webinar is being recorded, and access to the recording, as well as the PDF of the files, will be made available to you via email early next week. I'd like to introduce our first panelist, Todd Harrison. Todd is a partner at Venable Law Firm and focuses his practice on FDA, multiple divisions of the Department of Agriculture, and of course the FTC, just to name a few of the federal agencies he works with to support his clients' interest in federal, state, and the international arenas. As many of you know, Todd works extensively in the areas of food and dietary supplement safety, labeling, and advertising claims. His expertise in compliance issues is highly regarded, and he is passionate about the need for transparency, traceability, and accountability when it comes to the goods consumers consume, and the need for quality research that demonstrates how these products impact health status. Ergo, his role in the formation of the Organic and Natural Health Association and his service as our president, as well as his pursuit of our research agenda. Thank you, Todd, and I will now turn the presentation over to you.
Thank you, Erin. I want to thank everybody for joining this call. Many of you may already know that RCTs and clinical research has become a bit of a pet project of mine over the last year or so, especially in the last 3 months. This will be the fourth time I've talked on the issue of the need for a change paradigm when it comes to clinical research, and whether we should be looking at RCTs as being the gold standard for everything. I think from my perspective and from everybody who speaks here, I think that what we're looking to do is make the process better and to come up with a new way to address some of the challenges. No, we're never going to get rid of RCTs. No, would I suggest that we need to get rid of RCTs. But the word competent, reliable scientific evidence has many different meanings. And one of those meanings is other ways of looking at research, not just clinical studies. And that's what this is about, because nutrients are particularly difficult to study in the RCT environment. It would take 5 to 10 years of a clinical trial before you could probably even get to the point where you might make decisions about the nutrient. So I'm excited about this, and I look forward to going on with my presentation. I need to go to the next slide. There we go. So here are my consideration points for everyone today. First, it's time for a paradigm shift, and this is a policy issue. And it's a policy issue that requires everybody to start engaging, and that's just not engaging within the industry, but also engaging the regulators, because the regulators drive the policy, and they're stuck on RCTs. And so it's time to start educating them that there is a need of flexibility, that in fact the competent, reliable scientific evidence standard is flexible. Yes, we need an evidence-based model, but it does not have to be an inflexible model. It can be flexible. RCTs, by their nature, are inflexible, especially when you decide that everything has to reach a 95% confidence level and statistics override clinical relevancy or meaningfulness. So we can go to the next slide. So competent, reliable scientific evidence means basically just tests, analyses, research studies, or other evidence based on the expertise and professionals in the relevant area. that have been conducted and evaluated in an objective manner by persons qualified to do so, using procedures generally accepted in the profession to yield accurate and reliable results. So one of the questions I've always asked is, are RCTs the holy grail of life in clinical studies? And it's not. While they are a good element of it, the Seventh Circuit has explicitly held that nothing in the Federal Trade Commission Act requires placebo-controlled double-blind studies. Placebo-controlled double-blind testing is not a legal requirement for consumer products. It's probably news to the Federal Trade Commission. I appreciate that, but that really is what the law is. It's not. It is, to be sure, they're an important part of evidence, but they're not the only part of evidence. And what we've done recently in clinical study environments, and we've done in just research in general, is we're trying to create black line tests for things that, and take out any judgment. Well, if we all remember back from our history, back to the Greek civilization and the dawn of medicine, medicine used to be an art, not the science. And we're now taking RCTs take the art out of this, take art out of the science of medicine, and we can't do that. And that's why I'm a big believer in trying to change the paradigm. As former FTC Commissioner Bill Cloud said, when he criticized overzealous state agency and public interest groups advocate for absolute scientific certainty, he expressed a fear that under the line of analysis, the perfect could end up being the enemy of the good. And that's very true. Nothing's perfect. We cannot live in a society where we require no risk at all. And the fact of the matter is, when we wake up this morning thinking everything's going really good, and then what happened to me, I get in a bad car accident this morning. But yet you go on with your life. You cannot predict it. But yet that's what many people are trying to do to this industry and how to look at science. Statistical significance is an interesting issue. When you go back to it was first introduced to p-values back in 1920. It was intended simply to be an informal way to judge whether evidence was significant in the old-fashioned sense. It was intended to be a process that blended data and background knowledge, but it somehow got swept into a movement to make evidence-based decision-making as rigorous and objective as possible. Scientists who were not statisticians created a hybrid system that crammed Fisher's easy-to-calculate p-value into a rigorous rule-based system. This is when a p-value of 0.05 became enshrined as statistically significant. P-value was never meant to be used this way. It muddles thinking, and what's really worse, it's not relevant to whether there's practical relevance to a study. I learned that in a recent investigation where I had a statistician actually tell me that, well, even though you may not reach statistical significance, I never said that your data wasn't clinically relevant. Indeed, it was clinically relevant, and indeed, the product did work, which is very interesting. But yet, if you take that to the FDA or the Federal Trade Commission or a plaintiff attorney, because they like to sue people as well, they all scoff at you. We go to the next slide. So statistical versus clinical outcome. So what to do with clinical trials where positive clinical outcomes are observed but do not have statistical significance? Is there no value with data that shows a p-value of 0.05? Or what other methods are there to determine efficacy other than p-values? Shouldn't we be able to look at population-controlled studies and look at epidemiological studies or just plain observational studies as a whole to determine whether something's actually reaching and having effect? And could we have a situation where we do a long-term look at observational study and we show that people over 5-10 years are having a benefit, and maybe let's say cancer, for instance, and let's say we show that if people got enough vitamin D in their diet on a daily basis of 5 to 10,000 IU that you would decrease cancer deaths by 100,000 people a year. Well, and this is just an example, by the way. So, but you only had a p-value of 0.7. Well, I think those people, that 100,000 people who not have gotten cancer, would have thought that would be quite relevant to them and knowledge that they would have liked to have known. So it does, the whole idea of the way we look at trials and look at science doesn't make sense, because many experts would look at that and say, you know, there's a real effect here. And if people would actually start taking vitamin D, we may very well lower the risk, the cancer rate in this country, and have a lot of money saved on healthcare costs. So the question becomes what to do with these clinical trials, and what is the difference between the relevancy and the significance of these trials. So remember, statistical significance simply indicates the probability of incorrectly rejecting a true null hypothesis, never meant to be a rigid standard. It's actually kind of arbitrary number. We could have easily picked 85%. At a conference I was at and spoke at back in November, a statistician wrote that rarely does anybody ever repeat that 95% of the time is a study repeatable, which is actually very interesting. And, you know, again, it tells you that it's a difficult thing. The number really is arbitrary. It doesn't really mean it. Can a study fail to reach statistical but still be clinically relevant? Absolutely. As my story indicated, it can absolutely be clinically relevant. You know, you can be at 80% or 85% of p-value of 0.15 and still have clinical relevance, even though you don't reach the statistical relevance. Studies that are statistically non-significant are ignored these days because even though there's a true treatment effect. It could be due to small sample size. It could be the design of the study. It could be many different ways of looking at that study. Studies that show small differences, though, can actually reach statistical limit by increasing the number of subjects in a study. So if you're looking just to move the point just slightly, all you have to do is enroll enough people in that study to be able to reach statistical significance. Again, this is not for me to say that we shouldn't have RCTs. I think RCTs are a necessary part of all the paradigms out there that we look at, but it's not the only way of looking at science. Next slide, please. So one way that people have suggested that we look at it is look at something called minimal clinical importance difference, which attempts to define the smallest change in a treatment outcome that a patient would identify as important. Let's go back to my example of even if you don't reach statistical significance because everybody increased their vitamin D to 5 to 10,000 IU a day, but it did save an additional 100,000 people's lives. Well, yeah, maybe that's not statistical significance, but it's definitely clinically meaningful to those people to have that information in the first place. But all of this requires a paradigm shift. And without that paradigm shift, we're not going to get anywhere. And that paradigm shift has to start at the FDA, the Federal Trade Commission, and the halls of Congress. And we have to start initiating this debate, and it's a tough debate. But you know, anything that's worth doing is difficult. But sitting on our hands is not an answer to it. And so I really believe that it's time to sit down and engage everybody and listen to other alternatives of how we can substantiate claims, especially when it comes to nutrients. This will necessarily, the one thing that I'm going to ask everybody on this call, and I pointed out back in a couple of my other conversations I've had, is that do you really want me deciding what is clinically relevant and what is not? Because that's what you have at the Federal Trade Commission. You have a bunch of lawyers and economists deciding what is clinically relevant and what is not. They don't have a bunch of MDs and PhDs on staff. It's lawyers and it's economists. And the economists serve as statisticians in these cases. They're not the right people. Yes, they hire experts, but they're experts. They just like everybody else does. We all in the world of law, we hire experts that agree with us. So I think that why it's important that you get out there and start making a difference in how we make this change. You know, the fact of the matter is experts can always disagree on the clinical relevance of any clinical trial. But, you know, the bottom line here is that we have a First Amendment for a reason. And the First Amendment actually does allow us to communicate that, even if the Federal Trade Commission disagrees, even if the FDA disagrees, even if Department of Health and Human Services, NIH, CDC, they all can disagree with you, but you have a right to be able to tell people what is relevant as long as you fully disclose. So one paradigm shift that we've talked about in the past is the proof of efficacy to a proof of probable harm for dietary supplements. This is for nutrients, a shift in decision context be made from a proof of efficacy to that of probable harm. A calculus of benefit versus harm of the intervention should be evaluated on a nutrient-by-nutrient basis. Proof of harm of donor versus ready is already established in people who are not in a disease state but have parameters suggesting a disease. Without intervention, these people have high probability of developing disease. If the dietary supplement intervention can be demonstrated to be safe through high-quality and comprehensive safety studies, calculus of benefit versus harm of the intervention should shift towards the benefits of the supplementation. In the context of supplements, the placebo group represents no intervention. And I think this is probably a good time to switch the discussion to Dr. Haney.
Thank you, Todd. It's my pleasure to introduce Dr. Robert Haney of John Creighton University, Professor Emeritus and Professor of Medicine. He has worked for 60 years in the study of osteoporosis, vitamin D, and calcium physiology. He's the author of 3 books and has published over 400 papers, chapters, and reviews in scientific and educational fields. The major theme of his work has been quantitative physiology, portions of which you will hear today. Dr. Haney is actively engaged in nutritional policy issues. He has helped redefine the context for estimating nutrition requirements and has focused attention on the inadequacy of drug-based research designs for the evaluation of nutrient efficacy. Dr. Haney is widely acclaimed for the development of a set of standards for the evaluation of nutrient effects. Over his career, he has altered 2 basic paradigms of nutritional biology, showing 1, that nutrient deficiencies produce long latency disease as well as their classical acute disorders, and 2, that low bone mass was a marker for osteoporosis, not the main cause of its bone fragility. We are honored to welcome Dr. Haney today, who also serves as the Research Director for GrassrootsHealth. Dr. Haney.
Thank you, Karen. I'm going to be talking about some of the problems associated with designing nutrient studies, and to a substantial extent, some of the approaches that I shall describe will in fact be used in GrassrootsHealth, and you'll hear from Carole Baggerly to that effect in just a moment. Slide, please. Now, basically, there are 2 frameworks from which we can approach this issue of nutrient effects or the nutrient requirement. The first is risk assessment. And by risk assessment, I mean the prevention of some disease outcome. The alternative approach is to study the physiology and how the product is actually working in functional optimization within the body. These are 2 distinct approaches. The conceptual framework is fairly obvious. We start with the recognition that I think everybody shares, and that is that health is more than the simple absence of disease. Now, if we look at the continuum of intake from low to high, we see that at very low intakes we have classical diseases. And at the far end, when we've got enough of all the nutrients concerned, then we have full health. But in between those two, is physiological dysfunction. That is, the apparatus isn't working right. We don't have as much reserve. We don't have as much resiliency. We don't have a lot of things, but we don't have an explicitly recognizable chronic disease. And so you see, it takes a higher intake to ensure adequate function than it does simply to prevent disease. And I think many of us are really concerned with optimizing physiology and not just preventing disease. Now, if we take a physiological approach, there are several courses we might follow. For instance, this first one is the point of minimum required compensation. That is, how much does the body have to work in order to compensate for the fact that it's not getting enough of a particular nutrient? The second one is, what was the intake to which our physiology was adapted by natural selection? If we know that, then we have a pretty good fix on what it is we need today. The third is the plateau effect model, which is the intake needed adequately to support a critical function, such as calcium absorption and vitamin D, for example. The next one has to do with our reserves. It's the intake that provides maximum resiliency in the face of everyday stresses. How much reserve do we have? The next is the support of a critical function, and I'll show you an example of that in just a moment. Slide, please. Now, of this group of 6, we can identify 3, for instance, that are easily applicable to a nutrient such as vitamin D. Different vitamins or different nutrients will have different sets of requirements, and so these have to be tailor-made to the nutrient concerned. So a physiological requirement for vitamin D is the intake that calls for the least day-to-day adaptation or compensation. Secondly, it's the one that our bodies have been adapted to by natural selection over the millennia of human evolution. And finally, it's one that's needed to support one or more essential physiological functions. And we don't have time to go through all of these in this brief period this afternoon, so let's start with matching the ancestral intake. Here, humans evolved in equatorial East Africa, where we got sunshine all the time, every day, year-round. And, of course, we made a lot of vitamin D since we weren't wearing a lot of clothing, so we had a lot of exposed skin. 2 groups in particular have been studied in East Africa, the Masai, who are pastoralists and herd cattle, and the Hadza, who are hunter-gatherers. For the Masai, we know that their diet differs substantially from the ancestral because they live mostly on milk, the product of their cattle, of course. But their latitude, where they live, the skin, pigmentation, and so forth, all of those are entirely the way they were under ancestral conditions. Now, for the Hadza, however, which we see here, everything is the same as it was so far as we can tell under ancestral conditions. Diet, latitude, skin exposure, skin pigmentation, they're all the same. And anthropologists have, in fact, dubbed these people as the last of the first. That is the only remaining vestiges we have of our primitive forebears in the form in which they evolved. Now, when we measure vitamin D status in these individuals, and we do that by measuring a compound called 25-hydroxyvitamin D, which you see here on the vertical axis, what do we find in the Masai and the Hadza? Well, we find they're up around 110 to 115 nanomoles per liter. Well, what does that mean? Well, let's compare that with the Institute of Medicine number. It's right there. It's better than twice what the Institute of Medicine told us was enough for us to be fully healthy. The ancestral situation would suggest the IOM was low. That's one way to approach this issue. Next, I'm going to switch to the support of a critical physiological function. I think this is easier to see. And in this case, I'm talking about lactation, the mother feeding a baby with human breast milk. Now, there are a number of facts which you may or may not be fully aware of, and that is that human milk is really quite capable of providing all the nutrients, including all the vitamin D, cholecalciferol, that an infant might need. However, 25-hydroxy-D, which is how we measure vitamin D status in the mother, doesn't actually cross from the blood into milk, whereas the native vitamin D does. But that happens only if vitamin D is present in maternal blood. If it's all been converted to 25-hydroxy-D, then you won't have any D in the serum, and you won't be putting it into the milk. And unfortunately, for most of us today, serum vitamin D at prevailing intakes that we find in the industrialized nations, serum vitamin D is close to zero. 25-hydroxy-D may be higher, but not the D itself, and it's the D that the mother needs to put it into her milk. And that D concentration doesn't rise appreciably until the hepatic 25-hydroxylation, that is the step that converts D to 25-hydroxy-D, until that reaction is fully saturated. Second, human vitamin D concentration is in milk is about 1/3. That is, it's been reported in the range of 28 to 44%. About 1/3 the concentration that you find in the mother's serum. And if the mother's serum has zero, then 1/3 of zero isn't very much. Next, in order to meet the recommendations of the American Academy of Pediatrics for infants, that is 400 international units per day from breast milk, maternal serum D has to be about 12 nanograms per milliliter. That's a lot higher than most of us have today. At that serum D level, serum 25D, which is how we usually measure vitamin D status, would be about 50 nanograms per milliliter. And that requires a vitamin D input of about 5,000 to 6,000 international units per day from all sources, sun, food, supplements, whatever. However, at the Institute of Medicine figure for adequate 25-hydroxy-D level, that is 20 nanograms, no vitamin D gets into human breast milk. So the infants are deprived, and they must be dependent upon the administration of a vitamin D supplement. Now I'm going to go back to this disease prevention issue because if we don't take the physiological approach, we are stuck with the disease avoidance approach. And I'm going to switch to another nutrient entirely. But before I do that, I want to list the features that Karen spoke about, that I have written about, that are the requirements for a nutrient study to be informative. I'm going to run through these one at a time. The first one means study has to be done in deficient individuals. If you've got enough of a nutrient, giving more isn't going to produce any effect. So you've got to start out with people who don't have enough. You would think that's a no-brainer, but in fact, many of the studies of nutrients have ignored that one. Secondly, the change in intake must be large enough to change nutrient status meaningfully. If you're giving vitamin D, but it doesn't budge the serum 25D, then you haven't changed nutrient status. So you haven't given enough, basically. So, again, these things are common sense, but they've been so often ignored that's one of the reasons why we've had trouble with these kinds of studies. The next one, co-nutrient status has to be optimized. And what that means, next, please, excuse me. What that means is that the diet has to be fully adequate in all other nutrients. Nutrients work together like the instruments in a symphony orchestra. You can't do it with just one of them. If you've got consumers or patients who are deficient in 5 nutrients and you give them back only one of them, you may get no effect at all, or it may appear to go in the wrong direction. So if you want to see whether your nutrient is producing a benefit, you've got to be sure that the people receiving it are adequate in all other nutrient intakes. And without that, you will get confusing and perhaps contradictory results. Next, please. The bottom line is that if these requirements are not or cannot for some nutrients easily be met, then the resulting study may produce a null result, even if the nutrient is efficacious, or as Todd just said, even if it's clinically relevant. But you don't get the results if you haven't done the study right. So to wrap up, I hope I've shown you that disease prevention and health optimization are not the same. They're two different endpoints, two things you want to show. The latter, that is health optimization, requires a higher intake of a given nutrient than the former. A physiology-based approach, such as I've illustrated, a physiology-based approach to nutrient requirements is grounded in what a nutrient actually does in the body. It's designed to augment and facilitate normal physiological function, that's where we ought to be measuring nutrient effects. Finally, efficacy studies of disease prevention must meet certain well-defined but unfortunately often ignored conditions. And in that regard, let me remind you that the systematic reviews on which the Institute of Medicine and the U.S. Preventive Services Task Force in their reports in recent years have relied, those systematic reviews have been seriously flawed because they included most of the studies that they used did not meet these well-defined but often ignored conditions for an informative study of a nutrient. The result is that the aggregate average effect was generally close to zero because they included most studies that couldn't have shown anything even if they had wanted to. Thank you.
Thank you, Dr. Haney. Just remind our audience that if you do want to submit questions, it looks like we will have time to take those questions at the end of the presentations. Please use your chat to do so. It is now my pleasure to introduce our last presenter, Carole Baggerly. Carole is the founding director of GrassrootsHealth, a 501c3 nonprofit public health promotion organization with the direction of moving research into practice. Prior to forming GrassrootsHealth, Carole was the co-owner and vice president of sales and marketing for ESIS, a multi-million-dollar organization she founded in 1992 to help aerospace companies get greater operational efficiencies through electronic data interchange. And Carole's background in developing and running large data technology organizations has enabled the creation of a sophisticated data platform for the management of Grassroots Health data and the ability to create interfaces with major medical record systems. Carole has a degree in math and physics and has over 40 years of experience in consulting and business process reengineering and new business startups. Thank you, Carole. Please proceed.
Thank you very much. My objective with this presentation is to tie it all together. You've heard from Mr. Harrison and Dr. Heaney about what the problem is and certainly what the methodology solution is that Dr. Heaney presented. And our whole direction at Grassroots Health is to address those needs. Do you know how long it takes to get research into practice? Right now, it's currently estimated that it's 17 years or more, believe it or not, and that is too big a gap between the basic research and clinical practice. And before I go on from here, I want to talk a minute about, okay, fine, what are you going to do about that? The very first thing really is to assemble a group of scientists, an esteemed panel, as it were, who say, okay, what do we need to do? And what is the consensus of the scientists on what needs to be done? In 2007, I had the pleasure of attending a National Cancer Institute presentation on vitamin D and cancer. And after 2 days and probably more than 100 presentations, I was awed with all the information that was already out there telling people that vitamin D, and of course that was the topic, was absolutely beneficial to many situations, unknown to others. And at the end of the conference, one of the leaders of the conference said, where do we go from here? And his answer was, more research is needed. I gagged. And that literally was the formation of Grassroots Health. At that exact conference, after that conference was over, I had more than 100 of the researchers in that group come up to me and say, how can we help get the message out? And the researchers cared to make this happen. We then spent at least 6 months traveling the U.S. and Canada and now throughout the world to assemble a team of scientists who have made a consensus that everybody's serum level needs to be in the 40 to 60 nanogram per milliliter range. It is very exciting to take that message out. From that consensus statement of these scientists, Grassroots Health then designed all the pieces to make that happen. The very first thing that we did was to look at who are we going to be approaching with this. And the consumer-oriented group was really the one that we chose to go after, because at this point in time, with vitamin D and many nutrients, as you know, going directly to the medical profession is slow and arduous, whereas if you go to the general public, you will find great acceptance because they already want to know, what do I do? So we chose to do that. Another key component of this was a lot of hesitant people were asking, oh, but how do you know this, meaning the vitamin D supplementation is safe? Well, if we have a group of thousands of people doing what is recommended by the scientists, we also will be able to track safety, which we do. And I'm very happy to tell you, after 8 years in this process now, we have had 0 safety issues with people being at that level. The other thing that we do in this project is to track the various health outcomes and nutrient measures. They are quite documented. The nutrient measures actually is a vitamin D test. And as we move forward into doing other nutrient things, there will have to be supplemental tests, because you really want to know what is your vitamin D level and how it is related to the health outcomes. And then last on this list, but not at all least, is the outcomes of this research that we are doing is published, certainly in scientific journals, but please note the first comment to consumers. The people that are participating are actually the first recipients of what's happening. And it's what's happening to me, what's happening to my health, what's happening to the group of us who are participating. The Bridging the Gap that we have with our current D-Action project is internet-based. It's absolutely open to everyone around the world. The intervention of it is considered education. We provide lots of education, whether it be on a video or something, or just the very fact that people are getting a test result. That is educating them on what their supplement level is doing for their health. In addition to that, of course, capturing the health information and the things that are necessary to be able to publish and get results, reporting on what we call My Data, My Answers. This is the system that actually goes back to the consumers to give them what they want to do. And I want to also mention one comment here. When we first started this, I would personally get lots of, oh, thank you for doing this. And I would ask, what are you thanking me for? And the answer invariably was, thank you for letting me take charge of my health. And folks, this is all over the world. This isn't just the U.S. we're talking about. And then, of course, we also publish all of our information in peer-reviewed journals, and we have our publications in some very significant journals, from the American Public Health Journal to Cancer Registry information. A quick look at who participates. You'll see on this chart, the dark greens are where we have the most population right now. Certainly the U.S. and Canada are heavy, the lower half of the North Continent. You can see over on the right side quite a group in Australia, India, China even, and then up in the little green area up in the UK, we have quite a number of participants there as well. Our achievements to date, really significant successes that I think that we have had, is enrollment. People are still enrolling in the project, and we have more than 10,000 people worldwide. A major achievement is the health achievement. The recommended serum level is at least 40 nanograms per milliliter, and people have gotten there, all on their own, with education, with the feedback of how they are performing. And then certainly the education itself, the videos and things that we do. We have literally, at this time, millions of views of these and participation. And one of our specific charts, the disease incidence prevention chart, has actually been downloaded more than 10 times. And, of course, as mentioned, we have publications in peer-reviewed journals, which is very significant. I want to show you briefly some of the things that go out to the consumers and get published as part of what we are writing. Everybody wants to know, how much D do I take? What should I be taking? And this chart shows where a lot of people, where everybody is in the project. And that item where it has the arrow on it at 4,000, if somebody's taking 4,000 international units a day, somebody might be at 20 nanograms per milliliter, which is that little red dot at the bottom. Somebody might be at 120 nanograms per milliliter, which is that green dot at the top. The point of this chart, the information that goes with it, is that you have to test to know where you are and not give you a flat recommendation for what your D level is. On the next one, it says, how long do I stay in the sun to achieve a specific serum level? Well, if what you want in the sun is right at that 40 level, you see you almost achieve that right at the 30 to 44 minutes. So there's really no point in exposing your body to the sun for a longer period of time. Another one on a health issue. Can vitamin D prevent flu and cold? Well, look at those numbers. On that blue line there, it prevents 16% of cold, which is great, but take a look at the flu line, a full 50% prevention of flu. And this is something that is published already and can be shown, and people are encouraged to take care of their health in many ways. Another project that we have in process right this minute, and it needs about a year and a half before we can publish anything, might vitamin D prevent breast cancer? If you look at that right now, and this is after about 3 years out of a 5-year project, there are 80% fewer cases with a serum level of 50 nanograms per milliliter. So it looks highly likely that we will indeed be able to show, again, in this cohort, that happens. Another one, this other one I showed you on kidney stones, because there is a myth out there that says, oh my goodness, if I take more vitamin D, I'm going to get kidney stones. Not true. What we showed actually is that the lower people actually had more kidney stones than the higher people. The next chart is almost our most dominant comment chart. Can vitamin D help my pain? Well, what do you think? There is a very significant pain reduction. And as a matter of fact, when people enroll in the project, the very first comment we get very frequently after sometimes just barely weeks in the project is, oh my gosh, I thought I was going to have this pain the rest of my life and it's gone. So it is a very significant contributor to pain reduction. And then the last chart I have there, it doesn't matter what kind of supplement I take. Well, I think this is actually pertinent to the manufacturers as well, but essentially whether you're taking a gel cap, a liquid, or a powder, if you're taking the same amount, like 4,000 international units, it doesn't matter. You achieve the same serum level or the same average serum level. So you cannot say that one form of a supplement is better than another. What really matters is in order to take it on a regular basis. Okay, where do we go from here? So where we go from here are what I would call major themed projects. How do you put something together instead of just the mass population, which is working very well? We have now started what we call Protect Our Children Now. This is to demonstrate in a given community that the results, which have been published of a randomized trial, actually apply to a large community base. For those of you in the nutrient field, or in the medical field at all, you'll always hear, oh, that works there, or they did this study there, but we're here. Part of the purpose of this project is to demonstrate it works here, wherever here is too. We will be enrolling 500 women in a given community. We have just started a project in Charleston, which we are very excited about. Those women have to be between 12 and 17 weeks pregnant, and they will do vitamin D testing 3 times during their pregnancy. They will get supplementation to help them get to that 40 nanograms per ml. Their health outcomes are going to be measured, and the publication/promotion will be in about 24 months. Now, that public health promotion is an activist-oriented thing. I told a public health director in one city, I said, in 24 months, we are going to march on your office with 500 women telling you about what the benefits of vitamin D were. And God bless the lady. She says, why wait? I want to be involved now. So it's there. So we are starting that right this minute. Some future initiatives that we have coming and going certainly with organic and natural, there are multiple nutrients that we need to be looked at with their interactions. A lot of information is now out there about vitamin K, and we need to see what's going on in that interaction there. Additional population groups. We have many going right now, but I came in contact with another group this last week, such as elders. And they are a group of very astute people that want to make sure that our population of older people is adequately health conscious. And in particular, targeted marketing. Whether you are a distributor, a medical office, retail, what can you need, what do you need to do in order to expand nutrient health? So to get started, obviously, from anyone's standpoint out there or within the association, choosing a target group, an area of need, what benefit do you expect. Grassroots Health picks up the ball from there, essentially, to define the project and provide the quote for that project. And I mentioned earlier, but I want to reemphasize now, that Dr. Haney is our research director. And all of those points that he pointed out that are necessary for a quality nutrient study are what we do. And the funding itself for any study that we do is phased over the duration of the project. So from here, I wish you all help, and let's get this research into practice so that we can have healthier people. Thank you.
Thank you, Carole. We do have a few questions. Carole, I'm going to start with you. The first is, what is the time frame for successful conclusion of a population-based study, nutrient study?
Depends on what you're looking for. Our breast cancer project, which we have going, is a 5-year project, and that's due to a lot of statistical things, as it were, the size of the population we need and how infrequent breast cancer actually is in the population. This pregnancy study that we are doing, 2 years. So it really depends on what you're trying to achieve.
That's a really good lead into this next question then, is will this methodology work for measuring improvements in a disease state in a defined population, and what's an example of what Grassroots Health would consider in terms of taking that on?
Would it work? Absolutely. Because on the front end, you obviously do some evaluation of what those disease states are, and then you measure and track exactly as we do right now, what are the interventions of education and or supplements or nutrients of one kind. So absolutely.
Thank you. And Dr. Haney, okay, sorry, Carole,
let me
go ahead.
add quickly to that.
Sure.
There is a vitamin D deficiency epidemic around the world, and in one sense, I would consider that's the population we're already working with. Almost everybody is deficient, and consequently has many health outcome problems. Anyway, thank you.
Thank you. And that's a nice segue. Dr. Haney, I think this question is best directed to you. You referenced in your presentation that you can't measure just one nutrient in terms of being able to do a quality study. So how many nutrients, which nutrients, must be measured as a baseline for a study to be successful?
Well, it's not just a question of measuring. It's a question of knowing the nutritional context. For instance, if you're talking about calcium as a benefit for bone health, you've also got to recognize that bones are made not just of calcium, but they're made of protein as well. And you may not get a benefit of calcium if you haven't assured that the participants have an adequate intake of protein. Now, I cite that as just one example of how nutrients go together, but it's going to have to be specific for the nutrient that you are particularly interested in studying, and each one is different. So you need to be working with somebody who's broadly knowledgeable in the field of nutrition. Thank you.
A second question for you, Dr. Heaney. Can you speak to the role, to the issue of monitoring the impact of nutrients upon outcomes and any effect of prescription medications that enrollees are taking?
Well, obviously, prescription medications have an impact on total nutrition and could well alter how a person responds to a specific nutrient. For the most part, I think we're focusing on individuals who want to optimize their health. I quite agree it's very important to assure adequate nutrition to people who have chronic disease. There are two different issues, and so we need to be clear about separating them. Now, the first part of your question has already skipped my mind, so can you repeat it?
Well, it's essentially, I believe, how measuring the impact of nutrients on outcomes is impacted by the use of prescription medications in the study design.
Well, let's be specific in this case, and let's talk about, say, the control of blood pressure and sodium intake and drugs that lower blood pressure. Now, these things are all working in different directions, and in order to establish whether changing the nutrient intake, in this case sodium, is going to produce an effect, then you've got to be sure that everything else is the same. I'm not sure that I've adequately addressed that question, but I'm sure somebody's thinking about something very specific. And if that questioner can put that in terms of the concrete issues she is thinking about, perhaps I can answer it better.
All right, thank you very much.
May I add one comment there, please, Karen? There are a number of drugs that inhibit and or interfere with the absorption of vitamin D. So one of the things that needs to be tracked when you are going after some kind of nutrient adequacy, whether it be health or disease control, is to know what drugs that person is taking and what their impact is on the nutrient under consideration.
And just as a follow-up, Carole, all of that is done through your database and the patient intake?
I can't say all, but some.
Okay. Again, probably an issue for study design, I would assume.
You got it. You got it.
We have one while you're on, Carole. I think this is best suited for you. How do we change the paradigm with the medical community?
You have to be blessed with lots of patience. That's the calm kind, not the numbers of people that you see. But I think it's happening, Karen. It's you just have to. The world has changed. It has changed. That's an ED on the end of that. We have patients walking into their doctors this very day, and we did in 2007 when I did some of our initial tours, telling their doctor, I want a vitamin D test. And the doctors were saying at that point in time, huh? Now, and the patients were leaving. So the change that has happened is that we have a large segment of our population now who are adequately informed that they are helping that from the bottoms up. What's happening then is, like with this project in Charleston, we have a beautiful group of residents at the Medical University of South Carolina that are taking the lead. So it's happening. And we just need to do it more. We need to pick projects that are near and dear to people's hearts, like pregnancy is to everyone, and put together the projects in communities that say yes. And it's going to happen very fast, within the next 5 years.
Thank you. And Todd, the last question I have saved for you. Has there been any feedback from the FTC on the clinical relevance of patient-based research? And if not, how will we know they believe it's relevant for claim substantiation?
There hasn't been any feedback from the FTC. And the only time the FTC ever really wants to talk about clinical relevance, in our experience, is that when something reaches statistical significance and then they want to say, well, it's still not relevant. So the FTC is willing to use it as a dagger one way, but they're not, have not in the past really been interested in hearing, if you don't reach that 95% confidence level, why that study is still a legitimate study that supports your claims. And that's one of the crux of the issues we have. These are real policy decisions and policy matters, and it's really important that we start engaging the the agencies with it. Now, engaging the agencies have other issues because if you do a formal meeting or do a workshop with the FTC, they're going to invite all the consumer groups that will disagree with you, or invite conventional medicine that will disagree with you. So it's kind of a double-edged sword. But until we start having these conversations and start moving the agency, there's no reason for it to move.
Thank you very much. At this time, this will conclude our webinar. I want to thank all of you for joining us on our initial webinar of the year, and there will be many more like this to come over time from the Organic and Natural Health Association. I will also repeat that this webinar was recorded and you will have access to it to share with your colleagues, and we hope that you do so. Thank you. We appreciate your participation.